Okay, so good afternoon, everybody. I'll go ahead and get this started for those that are here. If anyone drops in, I'll just let them listen, but I will be posting this later. So welcome to the Unit 2 review. Here we go. Okay, so what was the purpose of this unit? So I think it's always good to go back and kind of think about what did we spend all of these weeks really learning? You know, it's very confusing, or sometimes we get very deep into what we're learning about physical control, chemical control, antibiotics, germ life cycles. Like, what does it all matter? Remember that this is Unit 2 is for control of microorganisms. Specifically, what we're looking for is we need to know how the microorganisms act, how they reproduce, how they infect others, what conditions are better for them to grow, what conditions help us defeat them better. And then by knowing that, we can create targeted solutions to try to attack these microbes with physical, chemical controls, and antibiotics. So you can see on the left, I like to put a little funny meme here. You know, you put 1 drop of hand sanitizer, we all know 99.9% of germs will die. But if you do 2 drops, theoretically, it should be over 100%. We know that's not true, but you know, it's a good way to like realize that we have all these different ways to treat to treat microbes that we have over the counter or that's prescribed. But we really don't understand how they're targeted. In order to do that, we have to kind of go through this chapter. So on the agenda today, we're going to go over nutrition and growth for microbes, genetics and reproduction. I'll kind of briefly discuss methods of control and antibiotics, which I think are your main takeaways for this unit. If nothing else, you should at least be able to understand the methods of control, both physical and chemical, along with the antibiotics in question. Okay, so just to recap the growth phases, we have our 4 growth phases. Initially, you have a lag phase where the number of microbes, I'll use bacteria as an example, are necessarily increasing. And then you get this log phase where they exponentially start increasing in number. Then they'll hit a stationary phase where they'll get to a point where every microbe that's in there is consuming all of the resources. And at one point, once those microbes are done performing their function and they reproduce, they'll start dying off exponentially as well. Different environmental conditions could affect our microbes. So remember, we have different conditions like temperature, oxygen, pH, and salt. I'm not going to go over all of them here. You should have them in your study guide, but I just put them here for quick reference. Understand the difference between a psychrophile, mesophile, thermophile, hyperthermophile, and what temperatures are associated with those types of microbes. And then oxygen. Recall we've had this discussion before, both in lecture and in lab. We have our obligate or strict aerobes. We have our strict or obligate anaerobes, facultative, aerotolerant, and microaerophile. I do expect there to be a question on this exam, specifically with a very similar picture like I have below, and it's asking you, based on what you see, what type of microbe could this be? I'm not saying it's going to be there, but I would expect something about like that to be on here. Same thing for temperature. You might be given a specific temperature, and you've got to decide what type of microbe it is based on temperature. Shrink this, get that out of the way. Again, pH, remember we have acidic, neutral, and basic, or acidophile, neutrophile, and alkalinophile. The pH associated with that. And then salts, you can have a halophile, which is about 15 to 30% salt concentration, or halotolerant, which is 0.1 to 20% salt concentration. Again, just review these. These are very good multiple choice questions. They might give you, hey, here's this pH, what type of microbe could it be? Or vice versa, they could ask you, hey, this microbe is an acidophile, what pH do you think would be surviving under these conditions? Remember, we have our 3 products of fermentation. This is very simple, but for some reason, this always sometimes gets missed or forgotten. So the 3 byproducts of fermentation, you have our yummy alcohol like wine, we've got our CO2 gas, and we have our acid. Again, this is a very good multiple choice question, a good listic question about, hey, click on the 3 or click on the byproducts fermentation. You should be able to easily say these three. I don't expect to be anything more in detail besides knowing the three byproducts of fermentation. Water movement, again, so you have hypertonic, hypotonic, and isotonic. What does it all mean? Basically, we have water inside of our cells, and we have water outside of our cells, and we have sodium or NaCl in our cells and out of our cells. Water will always follow the salt. So wherever there's more salt, the water will go out or in to make it even. So in a hypertonic solution, you have more sodium outside of the cell, so water will move out of the cell and the cell will shrink. Hypotonic, you have more sodium inside the cell, so water from the outside spaces will then come into the cell. Eventually, the cell will swell and eventually it will burst or lyse. An isotonic condition is the awesome balanced condition where sodium is equal on inside and outside. No cell size will change, and you also will not have any water movement. Okay, so genetics. We gotta remember that we have bacterial genetics, but we also have eukaryotic or viral genetics. For bacterial genetics, we're looking at primarily how do bacteria gain or potentially gain antibiotic resistance. So you have the three methods: conjugation, transformation, and transduction. Conjugation, always the big thing to remember is the sex pilus. So conjugation, sex pilus. If that's the only thing you remember, you'll probably be able to get it. Transformation is that we're using DNA from an outside source. Either we, as people, introduce this bacterial or this new DNA into the cells. That new DNA strands will combine with the original bacterial DNA, and then we'll create a brand new cell with both the original DNA and the new DNA. We can do this either as humans by inserting this to the cells directly, or in nature, you have cells that specifically go and hunt other cells. They'll destroy them, they'll take their genetic information, and they'll transform themselves by taking these other cells' genetic information. And the last one is transduction. Transduction is always done with a third-party bacteriophage, where the bacteriophage will steal the genetic information from one type of cell, and then it'll infect it into another cell, and it'll transform the cell that way. So again, just be able to know the difference between conjugation, transformation, and transduction. Question like this, if I was making a test, I would probably ask you, let me give you a definition of conjugation, transformation, and transduction and have you answer it, or vice versa, have you pick out a definition. This is also a very good drop-down question if you think about it, where I give you conjugation, transformation, transduction, and you have to drop down the ones that match. And then for viruses, we got to remember antigenic drift and antigenic shift. So remember that a drift is a small or minor change in viral spike protein. It's typically a very slow change, and it's caused by point mutations. Point mutations are just basically insertions or deletions of nucleotides, or a switch of a nucleotide in the DNA sequence, which causes the spiral spikes to kind of slightly change in what they look like or what they will attach to. But it's typically very slow over time, and overall the virus still looks very similar to the original virus. And then remember, an antigenic shift, you have a major change in viral spike protein. It typically happens very quickly, and it's caused by genetic recombination. Remember, we had the bird, the pig, the humans. All of these three types of flus or influenza viruses got into the pig, and inside the pig they all mixed, and it came out with a brand new virus that had viral spike proteins of both the pig, the bird, and the human. And now that new virus can infect birds, pigs, and humans, where before it could only infect pigs, for example. Okay, I think the biggest one that we're going to really, I kind of want to go over, I won't go over it too in depth, but this is one of those questions where, as a student, there would be certain questions or certain topics that I just kind of pushed off to the side and kind of hoped they would not be on the test. Methods of control. Oh, hi, do you have a question? No? Okay. If you do have a question, feel free to just throw it in the chat. Or you can also raise your hand and just say it out loud. But essentially, methods of control, I'm going to tell you it's going to be on the test. You're probably going to have about 4 or 5 questions on this, and they're going to be multiple like parts to these questions. So if you don't know methods of control, you don't know antibiotics, you're not going to do well on this exam, period. So this is one thing that if you only have time to study 1 or 2 things, study methods of control and study antibiotics. There's different ways to study this topic. The way I like to break it down is I like to list, very similar to how I did in this slide, is I'll go physical methods and sterilize. So what physical sterilants do I have? And I'll list them out. You can see I added autoclave, and including the pressure and temperature, UVC, and filtration. I did disinfection. I talked about boiling, pasteurization, and dry heat. There are other examples, but I just wrote a few. And then for chemical, I did all my chemical sterilants. Remember the O's, aldehydes, halogen, hydrogen peroxide. You should know the 3 O's, or sorry, you should know the 3 sterilants for chemicals. Like that's, I'm going to guarantee you're going to have a question on those. But then I also listed the disinfectants, and then you would also list the antiseptics. If you have time, I would either try to make something like flashcards, where you can say like, hey, what sterilizes physically? Autoclave. What sterilizes chemically? Aldehydes, 2% glutaraldehyde, for example. However it works for you, however you have to list them. If you want to just write all sterilants and put all physical and chemical sterilants in one list, you can do that. But if I was to make a question on this, I could ask you, I could give you a microbe, for example, and say, give me 1 physical method and 1 chemical method to sterilize. And then you have to write down those 2. And then I can ask you, give me 1 method to disinfect chemically and 1 method to disinfect physically. And that's 2 more things you have to write down. And then I can ask for, now give me 2 things that are good for antiseptics, 1 physical, 1 chemical. And you can see how I just built a question based on 1 question, how it could be worth 6 different points. So I ask you just 1 example for each to sterilize, disinfect, and for antisepsis. Page 7, if you do not have it completed, I would go back and make sure that it is completed. If you complete it, but you have questions, you can just email me and I'll review it and I'll give you feedback or make any corrections that you might have for that one. And then lastly, antibiotics, which is page 10 of your study guide. I don't know if you can see how I have it. I know we worked on it in class. That's how I broke it down here. If you have an example of this, just let me know and I'll send it to you. But basically what you want to know is the antibiotics target different things. So in your list or in your head, in your flashcards, you should have, all right, these are the drugs that list that attack the plasma membrane. These attack the peptidoglycan cell walls. These attack waxy cell walls. These target folic acid metabolism, also knowing which is more or less toxic. And then these target protein synthesis and also knowing which are more or less toxic, which target DNA and which are antivirals. We mentioned that the study guide is typically fair game. So every drug on the study guide is technically fair game. What I would tell you to do is try to create them in certain groups to where you don't have to remember every drug, but you can remember what some of those drugs do. And then that kind of creates the link where if you see the other drug, you know, like, oh, that's just like this other drug. For example, if I created a brand new drug called salsatocillin, for example, I would probably group it with all the other cillins like ampicillin, amoxicillin, methicillin, penicillin, which means that my salsatocillin drug would probably still target the cell wall of the peptidoglycan, the peptidoglycan cell wall. The mycins, streptomycin, neomycin, erythromycin, those all target protein synthesis. So if you know one of them, you can probably remember the other 2. Vancomycin is the only one that you have to be careful about because that one specifically targets the cell wall. And then polymyxin B is our only plasma membrane only thing that targets the plasma membrane. So I would definitely know polymyxin B, for example. I'm going to check out questions in the chat. Do we need to know the major site of action for a chemical control agent? I would say no. I think it's more important for you to know whether it's physical or chemical. So that'd be the first way I would break it down. And then knowing whether it sterilizes, disinfects, or is antiseptic. If you know those two, you'll probably get most of the tests. The other thing that you could be asked, especially for the chemicals, is what chemical groups do they belong to? So you could be asked a question about, give me a quat, for example, and that would be like 409 or fantastic, which is on page 7 of your study guide as well. Instead of if you're worried about trying to get all of them down, I'd really focus more on kind of like chapter 11 case study. I believe it was chapter 11, is we gave you a question and there was multiple correct answers. We just wanted you to give us a way to sterilize, a way to disinfect, and a way to have antiseptics. And if you in your head know, like, I want to use hydrogen peroxide 30% for my sterilization, I want to use 7% ethanol for my disinfectant, and I want to use Hibiclens for my antiseptic. If you know something along those lines, you'll probably do well on the exam. Again, you might get the onesie twosies that ask you for, give me an alcohol, give me a halogen. But the questions are going to be more tailored towards, here's this scenario. What are you going to do to sterilize, disinfect, antiseptic, both physically and or chemically? And that's kind of, I'll share this as, oh, does that answer your question, Yasani, by the way? Okay, perfect. And then the last thing I'm going to cover, because I'm running a little low on time. I only have 40 minutes for this meeting. I think it ends in like 5 minutes. But what I'm going to do is I'm going to throw a copy. Oh, that's the wrong one. So we had these practice case studies yesterday. We ideally ask everyone to kind of fill them out as a group. But I know we were kind of low on time. I'm going to throw that in the chat. If you want to take that and download it and then work on it yourself, the way this is structured, I hope you can see this screen. If not, I'm going to share it. Okay. So you can kind of see how this one's already structured, where you have, hey, I gave you a Mycobacterium tuberculosis in a surgical room. So it tells you, all right, identify that it's an acid-fast bacteria with a cell wall. If I know it's a cell wall, I only have 2 antibiotics that can treat it. And that's part of our page 10, all right? And so if you go back, you're like, all right, I recognize it's a waxy cell wall, so the only drugs I should be using are ethambutanol or isoniazid. I'm not really sure how to say that one, but it's on your study guide here. Imagine a similar question that we could give you a gram-positive bacteria. We could give you a virus, and you should be able to at least say, if I have a virus, I only have 3 antivirals. If I have a gram-positive bacteria, I probably want to target the peptidoglycan cell wall or the plasma membrane. Methods to sterilize, you can kind of see they use the same ones I chose for the examples for autoclaving, UVC, and filtration. And those are, again, physical methods of control to sterilize. And then you have physical methods to disinfect, chemical methods used to decontaminate, which is another way to say sterilize, high-level disinfectants, and then for antisepsis. You can kind of see how these cases ask you the same questions that I was asking earlier, and expect your exam to have questions very similar to this, where it asks you, give me some examples of sterilization, give me some examples of disinfectants, give me some examples of antiseptics, both physical and chemical. And it sounds like I'm saying that a lot. I'm saying that a lot because I'm telling you that's going to be on the test. a good chunk of your grade for this unit is those types of questions. So if you have not started looking at it, please, please, please focus on those topics. They're very important. You cannot get a good grade without knowing these topics. But yeah, I recommend just copying this, downloading it, and that way you can kind of delete these and work them out yourself, make some flashcards for them if you want to. I'll also send out the practice cases and the sterilants as an announcement, along with the video for this review session. That way you have that reference as well. Do we need to know if each, so I'm going to answer the question, do we need to know if each antibiotic is narrow or broad spectrum? I think it's important to know, but what I would say, it's not so much important that you know it, more so how to apply it. So I'll give you an example. So I said, talk about ethambutanol, right, which targets acid-fast, waxy cell walls. That's probably more of a narrow spectrum because I can only really use it on waxy cell wall or acid-fast bacteria. But if I gave you something like ampicillin, which targets the peptidoglycan cell wall, based on what we learned in Unit 1, gram-negative have peptidoglycan cell wall and gram-positive have peptidoglycan cell wall. And if a drug targets more than one microbe, it's broad spectrum. If it targets only one microbe, it's narrow spectrum. So one microbe, narrow spectrum, more than one, so 2 or more, it's broad spectrum. So you don't necessarily have to study it if you understand what the drugs are targeting. But if it helps you, you can definitely look it over. But I think it's more important to understand what the drugs target. And once you understand that, it'd be easier for you to say it's narrow or broad spectrum. Does that make sense? Okay, perfect. Great question. And then Darius or anyone else in the chat, if you have any other questions, you can type them in or just feel free to say something. Let's just stop sharing this one and go back and share the PowerPoint. Okay. So again, to recap, oh, here we go. Okay. So again, to recap, make sure that you have your study guide completed. If you've already gone to lab, would have gotten your extra credit points. If you're a part of the Wednesday group, we'll look at your study guide tomorrow, and I'll stamp those and you can get your 2 points. Again, everything on the study guide is fair game, but I would really encourage you to focus on pages 7 and 10 for the methods of control and the antibiotics. Create flashcards, create Quizlets, go and do some, what are those called? Those little like online like practice tests or practice quizzes that you can find. And that way you can at least get a good method of like trying to figure out what these drugs and what these methods of control target and what they're used for. But definitely understand, if we ask you for sterilants, know your sterilants, know your disinfectants, know your antiseptics, know which ones are physical, know which ones are chemical. And then for drugs, primarily know what drugs are used to target what are the targets of those drugs. And if I give you a type of bacteria, like a gram-positive bacteria, tuberculosis, influenza, know which drugs you're going to use to target those specific microbes. Last thing I'm going to leave you off with is trust yourself. You know more than you think you do. And like Ash and Pikachu would say, you can do it. This is not going to be that difficult of a test. I think if you know page 7 and page 10, most of the questions are going to come from that. Everything else is pretty straightforward. But I know coming off a break, it's always a little bit difficult to get refocused. I know I'm there as well. So just put in the time, put in the effort, get your flashcards done. The exam will open Thursday morning, and you'll have until Saturday night to complete it. You get one chance. It's going to be on Canvas, live proctored, or the Respondus lockdown browser. So if you took a Unit 1 and you were in a loud environment, you had iffy internet connection, I would try to make arrangements to come down to the library, or you can come to a study room. Like I'm in the library right now. I booked out a room for a few hours. I can just sit here, do what I need to do, be away from distractions, and then when I'm done, I can leave and again go take care of like my family, my dogs, your kids, go to work, whatever it might be. Do I have any other questions? Oh, there's something else in there. Okay, awesome. So if I have no other questions, again, feel free to reach out to me. I will have this posted tonight. It takes about an hour or so for this to kind of condense and get ready for it. But again, reach out if you have any questions, or if you're in the Wednesday group, you can see me tomorrow and ask me as well. Again, good luck, and I'll talk to you soon.